Hello, everybody. I'm taking Caddy's thunder just for a second. Please come back to your seats. We're going to start shortly. Just noting that this morning, Stats Canada came out with its yield results for the year. And canola's result was 44.7 bushels per acre, which is a Canadian record. The previous record was 43.2 in 2016. So that's, I guess, something to celebrate, and we can all give ourselves a pat on the back for our contribution to that effort. And I mean, well, genetics companies and farmers in particular, I'd like to point out. Anyway, this is for online too. Thanks, everybody.
Okay, good morning, everyone. My name is Kathy Navabi, and I'm a canola breeder. I will be chairing this session on the application of omics technologies in canola disease management. With me today, I have Jared on the online questions. So as you hear the talks, please put your questions in the chat, and he will make sure to capture them. Before we get into the program, I want to go a little off the script and recognize, as one of the members of the organizing committee, that this panel, this session this morning is not gender balanced. And it's important to me to bring it up because I want to make sure that it doesn't discourage or disappoint any of you young female researchers out there. It was an oversight, and we apologize for that. But going back to the program, our first speaker is David Halstead, a researcher at the School of Natural Resources and Built Environment at Saskatchewan Polytechnic, Prince Albert campus. The title of his talk is Early Detection of Clubroot in Canola Using Drone-Based Hyperspectral Imaging.
Thanks very much. I want to say, start off by saying, and now for something completely different. Take a little bit of a break from the genetics for a second here. Research I'm going to present here is a collaborative effort, including myself and Leila Ben Marouche, Research Associate with the Sustainability-Led Integrated Centers of Excellence at Saskatchewan Polytechnic, along with Dr. Bruce Gosson, Agriculture and Agri-Food Canada, and Dr. Mary Ruth McDonald, Ontario Agriculture College, University of Guelph. Now, I recognize that this is day 3 of a canola conference, so I'm not going to get into too much detail on this first slide. Just a quick overview of clubroot itself. Clubroot is an important constraint to canola production, obviously in Canada. The disease was first discovered north of Edmonton in 2003. It's easily spread between fields by contaminated farm equipment, as well as natural sources, wind, and so on. And early detection is difficult since above-ground clubroot tends to only become apparent after flowering when the crop density sort of prevents navigability, I guess. It's difficult to get through the field at flowering, put it that way. So recognizing that, we also have to recognize that detection is key to managing this disease. And the earlier the detection, the better. And so that's the problem that we're trying to solve here. And how do we go about that? Well, back in 2019, Sask Polytech was granted an Applied Research Technology and Instruments grant, and we used that money to purchase a hyperspectral drone to add to our fairly significant fleet of drones that we had at the time. And I was aware of the fact that hyperspec might be a solution to a problem that I'd come in contact with in talking to Doug Heath and others. and that was the clubroot problem. And Doug put me in touch with Dr. Bruce Gosson and Dr. Mary Ruth McDonald, and we sort of got together and decided that this might be worth an ADF grant. So we got together, put our heads together, and applied for an Agriculture Development Fund grant, and were awarded that grant the following year, 2020. And that grant then allowed us to pursue this research. Just a quick overview of hyperspectral. Hyperspectral sensors capture a continuous range of reflected electromagnetic energy in narrow wavelength bands. Okay, so it really allows us to discriminate objects very finely. The reflectance data can then be combined to produce unique spectral signatures that vary according to the composition and to some small extent the structure of the targeted item. Since clubroot is likely to affect compositional makeup of a canola plant, our hypothesis was simply that clubroot symptoms should be detectable by drone-mounted hyperspectral cameras. So as I said, we applied for a grant, we got the grant, and we started our research in 2021. Hyperspectral imagery was collected from 21 research and producer sites stretching across Alberta and Saskatchewan. So we wanted to make sure that we took in a very large geographic range of test sites. This is applied research, so we really want it to be useful on the ground. The sampling that we undertook, oh, pardon me. I see my notes now. When we did the hyperspectral sampling, the local crop specialists were invaluable for identifying candidate sites. And we also obtained written permission from all of the producers and research entities that we worked with. So there's no flying over somebody's field without their knowledge. Sampling efforts primarily concentrated at the field entrances, simply because that's where clubroot is most likely to occur or to be established. We also, of course, implemented biosecurity measures whenever we were on those properties so that we also did not aggravate the situation further. Spectral reflectance was captured using a headwall photonics nano hyperspec camera mounted on a DJI M600 drone. The nano hyperspec captures 273 spectral bands between 400 and 1,000 nanometer wavelengths. So you're taking that 400 to 1,000 nanometer wavelengths, and you're breaking it up into 300 or so discrete units, roughly two nanometers each. That's the discriminating power that I'm talking about. Slight changes in composition are going to show up with a slightly different spectral range. Drone flights were semi-autonomous, pre-programmed missions covering usually about a half a hectare to 4 hectares. And for those concerned about the changing incident light and things like that occur as you move from farm to farm, from year to year, every flight was preceded by calibration. We take into account the incident light by doing a white balance versus a dark balance. And we also had a calibration tarp situated inside every mission that we did. That calibration tarp would allow us to adjust the reflectance according to incident light conditions. You can see on the picture there, the map of Alberta and Saskatchewan shows the areas where our missions were concentrated. That's not showing all the missions, it's just showing where they were concentrated. And like I said, we really did count on local crop specialists to give us guidance as to where we might best find clubroot in those areas, if it existed at all. Okay, you also see Leela there down by the drone. That is the M600 Pro. And next to that, there's a small image of the camera that we use, the nano hyperspec. Again, it's a small camera, and it's situated on a gimbal so that it always maintains a directly vertical profile as it's looking down. And although it's a small camera, it has a pretty significant price tag. So this is where I'm going to tell you not to panic. This is the analysis workflow that we used, and I'm going to really try my best here to downplay some parts of this analysis because we would be here for a good portion of the day if I were to go all out here. Data collection and analysis involved one of two pathways. This isn't sort of how we initially envisioned this thing, but what happened is we ended up at the DLC research station west of Edmonton early in this project, and it completely changed our whole work plan. DLC has a fantastic workstation there, and lots and lots of different cultivars of canola, and of course they're doing a lot of clubroot investigations as well. We asked if we could fly their field. They said, yes, you can fly our field. They asked us not to disclose, of course, any of the cultivar information or anything like that, but I'm a natural resource guy. I'm not an agriculture guy. I wouldn't know what the heck they were talking about. But what I did want to know was, did they have a big diversity of cultivars? And of course they did. They were doing a lot of breeding and breeding testing. So for us, that was perfect. What I wanted to do was to get a wide sample of clubroot infected cultivars and non-infected cultivars to sort of homogenize the results of what we're trying to do here. We're trying to predict where the clubroot is and what effect the clubroot is having. I didn't want to pick up phenotypic differences between canola. So by having all of these in one place, that really helped us, and DLCs then became our training data site. So all of our training took place at that site. We collected a lot of data and processed that data, and then I subjected it to model development, as you'll see in a second here. The other data is showing at the top of that image, basically producer fields. We did about 20 producer fields across those two provinces, and within those producer fields, we would then take a smaller hyperspectral image, like I say, typically around the entrance, but extending a little bit into the field as well. And we'd pass that hyperspectral image on to Dr. Gosson and Dr. McDonald, and they would go and do the field pathology post-harvest. So that way we actually had the field data, and we also had this training data from the DLC orchard or breeding site. As it mentions there, the DLC research site was just perfect for doing this kind of analysis. And we use that as supervised classification data for later testing and validating the information we got from the test sites. The field validation itself pitted the best predictive classification model that we could derive from DLC information. And then we would compare that against the small patch data that Bruce and Mary Ruth would collect when they were out doing their field pathology work across Alberta and Saskatchewan. I should mention that when we did this validation and we applied our model against those small patch sites, we needed to receive a to label that with the clubroot designation. We needed to get more than 5% of the classified pixels displaying a greater than 50% probability of being clubroot infected. So I'm kind of skimming over the modeling part here, and I have to explain that there's almost like an entire second study in here, and it has to do with machine learning and using different machine learning tools to see which one would produce the better model for clubroot detection. And that's where I could sort of go off track, and I'm trying my best not to go off track. But to keep it short, we looked at stochastic gradient boosting, random forests, classification and regression trees, and binary logistic regression, which doesn't really fall into that machine learning area, but nevertheless was very helpful as well. So we did the comparison of those using the DLC test site data, and found out that well, I'm going to jump ahead to the results here. Found out that the stochastic gradient boosting provided the best machine learning result. We also found out from the DLC's data, because we could easily distinguish clubroot infected from non-infected thanks to their pathology reports that they supplied us at the end of their breeding season. We also found out that there is a very distinct difference, spectrally speaking, between clubroot infected and non-infected canola plants. Okay, and you can see that here on this spectral curve. The right side of the curve is the infrared information. Very big difference there on the infrared side, particularly in the 758 to 764 nanometer range. And then there's a slight difference down in the red area and around the 600, 650 range. And then another difference in the green part of the visual spectrum as well, around 550 range. So those differences really help to bring out where that clubroot is going to be and how it's going to show up. All right, so predictive model development, again, I sort of had to skip over this previously, but among the machine learning algorithms tested, again, that stochastic gradient boosting and binary logistic regression produced the most accurate classifications using independently assessed subsets of our training data from DLCs. So again, all of this is in the DLCs data specifically. We worked with, I better be careful. We worked with small sample subsets, 450 pixels each, and then I would apply those 450 pixel subsets against the others and compare and see if I could predict what was going on in each of those 450 pixel subsets using this model. And it turns out that the model was, when I got to the end of this process, the model was 96% accurate in classifying clubroot or non-clubroot situation, okay? So very, very good model, but I'm using training data here. I'm comparing the model against the training data that was used to develop it. So 96% is good, but you kind of expect it to be a pretty good performance when you're using the training data. All that training data then was subsequently then combined into a new 31 spectral band predictive classification model for field testing purposes. Okay, so at this point, these 31, geez, I can see the time's going here. At this point, the model now is in existence, and we're going to go and try it against field data. And we matched it up with a blind assortment of small patch locations correctly identified, and we correctly identified clubroot status at more than 90% of those small patch locations. Okay, so better than 90% predictive accuracy in those farm fields between Alberta and Saskatchewan, and collected over 3 years of data collection. Okay, all of that which took place during COVID, incidentally. So really good results. If I reduce that to just the field itself, we had 12 different fields involved in that comparison. We would have 100% accuracy at the field level for detecting clubroot. So we have a model that works. So the question is, how do we apply that model? Why don't we go ahead and just see if we can map clubroot on a field, just basically using our model. So that's what we're doing here with supervised classification. This is a hyperspectral image of a field in northern Alberta. You can see there's some areas there where there's some dead patches. We knew that this field had clubroot, so it's not a surprise, but we want to see if our model picks it up, right? So you can see that area there that's little bit bare. I removed the RGB, the red, green, blue portion of this image, so that we could see better what happens when we apply our model. And then we applied the model. I didn't apply it to the whole area, but I applied it to a large part of the area. And there you go. There's the club root, according to our model. The entrance to this field is in the lower right-hand corner of this image. I purposely left the entrance out because it's an identifier for the producer. I didn't want the producer to be identified. But the entrance is in that lower right-hand corner, so you can see that the club root starts to concentrate as you move towards that entrance, or as you move, as you're coming in the entrance, of course. There's something else that's really jumping out there, and I wonder if you're all picking up on it or not. The top part of that image, there's a lot of parallel lines of club root. You picking up on that? Those parallel lines are 25 centimeters apart. That's 10 inches. Turns out 10 inches is the average distance between tillage discs. So you can see those lines, those parallel lines. That could be the smoking gun of the contamination on this site. We've got that, and then aside that, there's that bare area that I pointed to before. There's nothing growing in that bare area, as we would expect, or very little anyway. So it looks to me like this model is working. We did pick up the club root we expected to see, and we're also seeing, signs of contamination from heavy equipment. So by way of discussion, canola plants were infected with club root, and canola plants that are infected with club root provide a distinctive spectral signature that can be used to identify infected patches within a crop. Remotely piloted aircraft systems offer an excellent platform for mid to late season club root detection, thus paving the way for more effective management. Among the various machine learning approaches used, again, stochastic gradient boosting provided the best overall result. Performance is borne out by the field validation results spanning multiple growing seasons, hundreds of kilometers of geographical area, and highly variable field conditions. Success of the study is owing to an adaptive approach to model development, the power of machine learning, and the quality of training and anonymized pathology data obtained from DLCs. If you want more information, I can assure you that it's going to be a lot more there. If you look at our paper, just published in 2025 here, this month or two ago, and the DOI is there for you. And I also want to acknowledge Dr. Stephen Fox and Glenn Hawkins of DL Seeds for giving us access to the anonymized club root ratings from their research site, and also allowing us to fly the site. Keith Gabbard of the Canola Council of Canada, or at least was from the Canola Council of Canada, for his assistance in finding fields to assess, especially in southern Alberta there, or Red Deer area. And also the numerous producers and cooperators that assisted with field sampling activities. Funding, again, generously provided by Saskatchewan Agriculture Development Fund and Sask Canola, as they were known then. Thank you.
Thank you very much. We are running behind, but we have 2 questions. We have time for 1 question. Oh my God, there are too many questions here. There is one online, and then we can ask if we can get to one from the audience.
All right, so this one's asking about timing. How early in the season can you detect spectral difference between club root infected and non-infected? And a bonus question, can green bridge infected weeds become identified as well? So alternate hosts for club root. Right.
So all the fields that we sampled with the hyperspectral camera were just in early to late flowering. So during that flowering period, I can't say for sure. I haven't gone back and looked at the actual dates of each one that we actually verified or validated on. But it's a great question, and I will take that up for sure. Thank you. I know there's going to be a lot of questions.
Great talk.
Yes.
The question is, like, is your model just comparing a yes/no, like type of analysis to yes/no, or is it quantifying the amount of club root?
Yeah, we worked with a binary data set, but the model itself provides probabilities as a product. So yes, we worked with binary data, yes, no, but as a result of the model, it will give me a probability. And like I say, you know, to say that there's club root there, I needed more than 50% probability from 5% of the pixels.
Thanks.
Yeah.
Okay, just to catch up with time, I'd like to encourage you to talk to David during the lunch break, and we will move on with the program. Thank you very much, David. Okay, our next speaker is Dinesh Adhikary. He's a research associate with the University of Alberta, Department of Agriculture, Food and Nutritional Sciences. The topic of his talk is integrative omics and microscopy approach to understand the potential cell wall remodeling in Brassica napus, underlying resistance against clubroot disease.
Good morning. First of all, thank you so much for the opportunity. Thank you, the organizing team. It's been going really well. Also, thank you, the tech team. I think it's a lot of work. Thank you for your hard work. Also, for the last few days, this is my first time with Canola Week, and it's been a very unique experience, and also very enriching for me, lots of learning. So today, my talk would be focusing on the kind of omics plus microscopy, but really, you know, our team at U Alberta is focusing on multiple traits, not only disease. Our team is fairly large. We have Dr. Rahman leading the group, and also we have Dr. Cave, and we have a few other PhDs, master students working on the projects. So really, you know, we are focusing on the basic and applied sciences, and really helping the farmers and the breeders designing markers. So today I'll be giving you some overview of my work for the last few years. So really, you know, I don't need to talk about this part, but what I like to really, you know, make a point is that as a researcher, our job is to make our farmer growers a bit easier with growing crops, right? So that means, you know, working closely with the farmers, growers, and breeders, understanding their needs and real challenges in the field is very important, and using the solid evidence-based data to help improve production and the profitability. That's what we discussed for the last few days. So I think as a researcher, it's also a very good, big responsibility for me. So to support and strengthen the program. So the important crop, canola, there are several challenges, and one of many challenges is the disease. And one of many diseases is the clubroot. So as you know, it's a disease almost like a pandemic, because we have the disease in more than 80 countries globally. So one thing that's very unique about the disease is that the disease can remain in soil for a long time. Even though if you don't notice the disease last year or this year in the field, that doesn't mean it is not there. It may be there. So from the management perspective, it's very important that you focus on the pH, soil pH, when it changes, also the moisture. Also, when you change the genotype, the pathotype can again thrive. So it's very important to make note of those points on the management side. So for the last few decades, well, I guess many decades, we have been working on the disease. We know quite a bit of pathogen biology. Also, you know, we have QTLs, genes related to the pathogen. So what's really is lacking in our field right now is the molecular understanding of how the pathogen and the host really interact. So that really brings to my question today is cell wall reinforcement is one of the strategies Brassica napus or the Brassica really reinforce to gain the resistance. So again, our program is really designed to support the growers. We integrate basic science, applied science, gene discovery part, design the markers, and then produce the new varieties for the breeders and the growers. That's our primary goal. But today, my talk is basically focused on the basic part, the discovery part. So in our program, I apply multi-omics, transcriptome, proteome, metabolome, combine the data set, gene discovery, and then so we apply the functional genetics to validate the genes, and then design the markers, and then apply into breeding program. So in my studies for the last few years, we have been using double haploid lines, Niels lines, to characterize the pathogenesis process. So here you can see we have the resistant genotype and the susceptible genotype. So we track the disease from day 1 all the way to day 35. So day 1, there is infection within 24 hours of inoculation. So it's a very rapid process. So we track the disease in both the genotype. And then so what's interesting is that in the resistant line, you don't see much change after 7 days. They look fairly similar. Those pink dots, pink matrix in the cell is basically the pathogen infected cells. On the susceptible line, however, you can see that after 14 days, you see a massive change. There's the evolution of the pathogen, or let's say like it's evolving in the process. So the question I'm asking is, what kind of genes, proteins, or the chemicals are produced by the resistant line to make it resistant and the pathogen not evolving in the process? So to understand the question I have, we have basically looked into multiple omics. So this is the transcriptome level changes we looked at. So we basically took a rutabaga line, double haploid, resistant line, and the susceptible line. We took the transcriptome and then compared the transcriptome. In the transcriptome data set, we got quite a bit of a response. So one thing that really stood out was the lignin. So as you know, lignin is a phenolic polymer that's almost like a physical barrier for the pathogen to get into the cell. So we saw quite a bit of gene transcript highly upregulated in response to the pathogen. And also, other thing we noticed was that the calcium. So last few days back, there was a poster on CPK28. So we also noticed like quite a bit of calcium-related genes involved with the calmodulin binding genes, which is involved with the cell wall synthesis process, directly, indirectly. And also, we noticed quite a bit of sucrose-related genes, like sucrose 1, 2, 3. So those genes are also involved with the cell wall biosynthesis processes. So, and again, so this study was based on the early stage infection where there is no gall presence. Now, similarly, we also looked at the later stage, which is the 7 days, 14 days, and 21 days. That's when there is a gall, like prominent gall, as you can see there. So here we took the proteome approach. And in our proteome approach, we were comparing between the resistant line, there is no gall, and the susceptible line, where there's a bigger gall, which is usually after 21 days or so. So it depends on the genotype. Sometimes we see the galls even like 17 days or so. But by 21 days, it's very obvious there is a bigger gall. So when you compare those two genotypes, in this case, those two lines, again, so we saw quite a bit of genes, proteins involved with lignin showed up. Again, the calcium showed up. Hormones, also there are quite a bit of genes involved in the process. So basically, there's a consistency between the studies between early days and the later days, but uniquely, genes involved with cell wall was highly distinctly upregulated in both the processes. So also, we did a combined approach. We combined the transcriptome and the proteome, and we saw quite a few genes involved in the process. Like here, you can see Kel's motif, peroxidases. There are remourins, which is also involved with cell wall biosynthesis process, highly upregulated, and they're consistent between the transcriptome and the proteome. So I'm sure if you're working on the molecular part, you're familiar with chromosome 3 and the chromosome 8. So depending on what genotype or what species you derive the resistance from, Brassica rapa, or the rutabaga, or Brassica oleracea, in most cases, we have seen the genes and the QTLs are housed in those two chromosomes or linked to those two chromosomes, especially in the chromosome 3, which are quite a bit of genes involved with resistance and also cell wall biosynthesis. So in our recent study, what we did was we took about 100 genes that are linked to chromosome 3, and we characterize how chromosome 3 is involved in the resistance mechanism process. So here, we developed the Niels lines, or the neurarsenic lines for Brassica napus. So on the right, you can see there is a 28 days after infection. It's a resistant line. There's no infection going on. On the other side, you can see the neurarsenic line for the resistance trait, where there is a bigger gall. So, and then for the resistance trait, it was contributed by chromosome 3. And then so when you compare between those two lines, we noticed that majority of variation was coming out from chromosome 3. More than 16,000 SNPs were coming out from that particular chromosome. And there are a few from chromosome 1, but mostly those genes involved with cell wall biosynthesis, and the resistance was in chromosome 3. So based on that, chromosome 3 is contributing quite a bit in this particular line for resistance. And then so when we did the transcript level assessment, we noticed that genes involved with resistance were highly upregulated in response to the pathogen. And also we noticed that there are several genes involved with cell wall. Like for example, here we have cellulose synthase upregulated. There's also we have the pectin-related genes, and also rhodesian-like proteins. And those are very much involved in cell wall plasticity, and also the lignin-associated resistance. So based on those molecular signals, we think there is cell wall reinforcement going on in response to the pathogen. So also to kind of dig more in detail, we use the Fourier transform infrared technique to get a molecular or the chemical level sense of how is that going on in those two genotypes or in those two lines. So we noticed that when you compare between the resistant anneal tissues versus the susceptible tissues, we noticed that there's quite a bit of molecular signals or the chemical signals that are linked with cell wall related components. Like for example, we saw polysaccharide-related functional group, lignin-related functional group, and as you know, lignin again is a phenolic polymer, which is a physical barrier. So having seen those functional group linked with those lignin and other components, kind of gave a clear idea that there's a highly likely chance that cell wall is reinforced in this particular line in response to that disease. So to dig more in detail, so now we want to see how is that in the cell wall. So we look at the TEM images, and we profile the changes that's happening in the cell wall in response to the pathogen. As you can see on the right, there is lots of those dots. That's basically is the pathogen cells. On the right, you can see those clean cells. In fact, it's a resistant line, but it is also not immune to the disease. You get the infection in the early days, but it won't carry to all the way to the galls, so which is a good part. So what you noticed was that when you compare into this 21 days sample, which is lots of galls at that point, so we noticed that by 21 days, the susceptible line has a thicker wall. It's almost like a swollen wall. And also the cell wall was degraded somehow. Whereas in the case of the resistant line, the cell was fairly intact and fairly smooth. So it almost gave an impression that somehow the cell wall is degrading in the susceptible line. And also in the 28 days samples, we saw very much similar pattern. The cell wall was degrading in the susceptible line. The cell overall was swollen, which we expect to see at that point anyways. But also the cell wall is highly degraded in terms of the susceptible line, whereas in the resistant line, it is much better. I mean, it's fairly intact. So from this study, so one thing that is very much relevant, I think, for this audience is that, Brassica family, there are more than 4,000 members within that family. Practically, majority of the members within that family are not, they are not immune to the disease. They get infection, they're resistant to not getting the galls, but they're not immune. So that brings to a point that because they can get infected, there's a highly likely chance that the pathogen can break the resistance anytime because there's a molecular interaction going on in the process. So that's probably why we are seeing that there is a quick breaking of the resistance in some of those lines that are resistant in the past. Also, other thing is that identifying those targets or the signals in the early days is very important, because what we saw in the greenhouse condition is that even though the line get infected, when there is no gall, the plant is still performing well, at least in the greenhouse condition. So having those genes, molecular signals identified, and if you can find the markers and design the molecular markers, and based on that, if you can apply into breeding, there is also benefit. Now the big question, right? So what does it mean to all of our growers? So I think this kind of studies, although it's basic, but it is very much linked to our applied sciences, because we're talking about markers for the breeders and for the growers. So this study will build stronger and durable clobber resistance. That's what we need. So by targeting cell wall-related genes. Also, it supports faster development of improved resistant hybrids, because we may not see the clobber this year or next year, but it's very important we have to continue producing those resistant cultivar or the resistant genetic material more and more, because you never know what's in that soil, right? Because the pathogen is evolving, and we saw that already in our studies. So this study also gives grower better hybrid selection process for the field management to make the field management decisions based on resistance biology. So finally, this is highly collaborative work. We have been getting support from lots of people, different institutes. Dr. Chaudhary from GIFS. We have Dr. Norton from U Alberta, Urmila Basu from our genomics unit, and of course our team, and of course the Canola Week. Thank you for this wonderful conference and experience. And our funders, AAFC, Ardor, Canola, Saskanola, Manitoba canola growers. Thank you.
Thank you, Dinesh. You saved us 3 minutes. Okay. So we have plenty of time for questions. I know there is one online, and then you go ahead. Yes.
Thank you, Adhikary. Excellent presentation. Yeah, excellent presentation. Yeah, very clear message, but in the new direction. So I know the Habib's labs, they are working a lot of resistant genes. Yeah. Now we are also targeting the cell-related genes. So do you see that resistant gene and the cell-regulated gene, do you compare that which one is like we already have A3 gene, A8 gene, yeah?
Yes.
So did you find any difference that cell-regulated gene and disease-resistant gene in the A3 and A8, there is any difference?
We found quite a bit of signals. Cell-related genes, of course, yes, we have quite a bit, like I showed in that slide also. So in fact, in the recent study, which I showed those results, based on 108 genes just from the chromosome A03. So those targets actually came from our previous studies. So we did multiple omics. We published in 2022, 23, 25. NEALS, double haploid studies. We have thousands of targets. Out of those targets, we selected more than 108 genes just from the chromosome 3. And also, as you mentioned that, within chromosome 3, there is a specific region, from chromosome position 20 to 27 MBS. So our Suman Raju also did some work on that part. So between that specific region in the chromosome, 20 to 27, there are specific genes involved with those resistance process. Yes, we have seen those things in our studies.
Okay, yeah, thank you very much.
All right. So we got one online saying, can synchrotron help decipher changes in cell wall structure? I think this is referring to Canadian light source imaging.
Thank you. Yeah, I'm sure, if you can use that facility, of course. That'll be really good, actually. We can think about that, maybe collaborate. We don't have those at U of A, at least I don't know. But we can definitely think about that. We could do as TM. That's the best we could do at this point. Thank you for that question. Thanks.
Thank you, Dinesh, for the excellent talk and your passion in it. Our next speaker is Mosammad Hur Madina. He's a postdoctoral researcher at Agriculture and Agri-Food Canada in Saskatoon. The topic of his talk is high-throughput functional assay of defense-related genes in response to clubroot.
Okay. Yeah. Hello. Thank you for the kind introduction and for inviting me to give a talk. Today I'll be presenting on one of my ongoing projects where I'm investigating the functional role of plant defense and immunity-related genes in response to clubroot. So as we have learned throughout the events and from previous speaker about clubroot and its economical impact on canola, so I will skip these introductory slides and I'll move directly to the major issue. So why managing this disease is so challenging? So we know so far we have the most effective management method is genetic resistance. However, the problem is rapid breakdown of this resistance due to fast evolution of new pathotype. And on top of that, most likely to have argine redundancy in canola. So it means that as we think, we don't have so many unique argines that we thought, and that's why resistance keeps failing. So now this also tell us that we need another approach besides the argines. Okay. If argine is not enough, so next question comes, how else plants defend themselves against pathogens? So we know there are two major immunity they have used. First one is PTI, so it's recognized pathogens, conserved molecules, and the second layer is called ETI. So this immunity uses our protein to detect pathogen-specific protein that is called effector. And both of these layers ultimately activate downstream defense response and including many hormonal pathways such as salicylic acid, jasmonic acid, and so on. So although many of these downstream signaling pathway has been well studied and effectively engineered against many pathogens, however, there is not much information about these genes against clubroot. So, one of the major advantages of exploring these downstream defense genes is to have broad spectrum resistance. So that was one of our main goals. So the funding, we have this work from Agriculture Funding Consortium for two objectives. So first one is gene mapping of resistance genes, and second objective was exploring downstream signaling pathways for clubroot. So today I'll be talking about the second objectives. So to explore downstream signaling gene, at first we have selected. So we have selected around 50 candidate gene based on our RNA-seq and published data. And most of these targeted genes are involved in the following pathways. So today I'll be focus on a few genes. For example, NPAR1. NPAR1 is well-known salicylic regulators. We know that means this turn on the salicylic defense response. And NPAR, and because Since NPR1 boost resistance, so this is, we have selected for overexpression candidate. Whereas NPR3, NPR4, and DMR6, it's act as a negative regulator. And that means it's turn off the salicylic defense. And it's, particularly DMR6 is one of the important defense genes because it has been shown that it is effective susceptibility gene against many pathogens. Since those three genes reduce or suppress plant immunity, therefore we have selected them for a silencing candidate. I'll also talk about some sugar transporter switch genes. During infection, pathogen actually hijack these switch genes and then export sugar for their own benefit. Since pathogen use these switch genes for their own benefit, we have selected them for silencing candidate. So there is also some nutrient uptake gene. So this gene particularly, it came directly from our RNA-seq data. And during clubroot infection, it was highly induced. So it is mostly likely to be excellent susceptibility gene candidates. Since this work is ongoing, so here we have named them gene A, B, C, D. So now we have candidate genes, but as we have so many candidate genes, so we are looking for some rapid and high throughput method. We know viral-based functional assay has been used as a rapid and high throughput method to study gene function for the model plant Arabidopsis. And this method sometimes it's not applicable for all the plant species, and we did not have such method for Brassica. So, but our collaborator, Dr. Kevin Rodowski, he has developed one for specifically for Brassica. So this virus system, he provided us to use it, and we have used this system for two approaches. One is for gene silencing, and another is for gene overexpression for our target gene. So many of us, we already know how the VIG system works. So in brief, so we cloned a small fragment of our target gene into viral vector and then transformed into the plant cell. Once it's in the plant cell, inside the plant cell process this fragment and then break it down into a small RNA, interference RNA through the plant RNA machinery, and ultimately it break down the native target mRNA, and it causes transcriptional silencing of our target gene. So on the other hand, for the overexpression, we cloned the full length of cDNA along with RNA silencing suppressor, which prevents RNA plant silencing and allows high overexpression of the target genes. So after having those construct of our target gene in viral vector, we have transformed them in the clubroot susceptible Brassica nepas line using agrobacterium, and then we have inoculated them with virulent clubroot spore, and after 4 to 6 weeks, we have scored the development of gall using 0 to 3 scale. So 0 means there was no gall, and 3 means there was severe gall and acid disease severity. So to confirm our virus system works, we have used two different markers. For overexpression, we have used PAP1 gene. So overexpression of this gene, it's produced purple phenotype on the leaves and roots. And for the VIGs, we have used PDS, VIGs control, we have used PDS. So this silencing these genes can produce bleaching phenotype. Okay, so now results part. So first we have tested NPAR1 gene. Since Arabidopsis NPAR1 is well studied, so we have used both Arabidopsis NPAR1 and Brassica NPAR1. And here you can see we have used wild type and empty vector as a negative control, and Brassica PAP1 as a positive selection marker. And for each construct, we have used more than 30 plants, and all this experiment at least repeated three times. So from the picture, image, and graph, you can see that all those control, negative control and positive selection marker was highly susceptible to the clubroot, and it produced big galls. And where, on the other hand, when plants expressing NPAR1, Arabidopsis NPAR1 and Brassica NPAR1, it significantly reduced the gall formation. And the DI value, it went down from wild type 90% to less than 20%. So this result suggests that NPAR1 is effective for the clubroot. So then we asked the opposite questions, like what if we silence the NPAR3 and DMR6 as they are negative regulators. So here we have used similarly wild type and empty vector as a negative control, and BNPDS as a positive selection marker. And similarly, these control also were highly susceptible against clubroot, and the plant silencing NPAR3 and 4 and DMR6, it showed significant resistance against clubroot. So this is really interesting. However, so this result suggests that NPAR3, 4, and DMR6 can be used, it's act as a susceptibility gene. And when we started this work, there was no information about DMR6, but recently one group has published that DMR6, silencing of DMR6 in Brassica oleracea, it can be effective against clubroot. So, but still, so this finding supports our results, and still nothing was known in Brassica nepas. So then we silenced some sugar transporter genes. So in Brassica, there is 17 isoform of sweet genes. So we have individually silenced them, and you can see from the image and graph, several sweet genes was silencing of several sweet gene. It reduced the gall formation of clubroot. And most effective one was sweet 4, 6, 8, 6, 7, 8, and 11. So the sweet 11 is, has been reported to be effective in Arabidopsis. However, to our knowledge, this is the first example of high throughput functional validation for all the sweet gene in Brassica. And next, we have tested the nutrient uptake genes, and here you can see, so similar in the all the control, it was highly susceptible, but for all 4 genes, from nutrient uptake genes gave high reduction of gall formation against clubroot. So the exact role of these is still under investigation. However, the initial result shows it can act as susceptibility factor against clubroot. So now we have all the clubroot phenotype data, so we wanted to validate this data by looking at their expression level using ddPCR. So in VIGS, we have seen that the gene expression was highly reduced, and in the overexpression, the target gene was increased. So at this point, we have validated most of the genes, but still work is ongoing. So here I'm showing an example for two genes. One is NPR34 and another one is DMR6. So as you can see, the reference gene, the housekeeping gene, was expressing consistently across the plants, and compared to wild type and the empty vector, the NPR3 and DMR6 was strongly reduced. So these results confirm that the resistance phenotype we have seen from the previous experiment, this is because of actual effect of the silencing of our target gene. So based on our VIGS result, we have selected most effective one, and then we developed CRISPR knockout and overexpression Brassica transgenic lines. So we have already confirmed for overexpression gene, we are screening T0 and T1 generation for single insertion homozygous line, and for CRISPR knockout line, we have done initially PCR and sequencing confirmation. We have some successful edit, but still we need to proceed for full confirmation for this mutation. And once we have those homozygous lines and knockout confirmation, we'll subject them for clubroot phenotype test. So as a conclusion, to our knowledge, this is the most comprehensive assay to study downstream signaling, downstream defense genes against clubroot. And through this pipeline, we have determined function of 26 genes out of 50 candidate defense genes against clubroot, and those identified effective defense genes we can use could be for manipulation to make clubroot resistance. And we have also generated Brassica, Nepas, CRISPR knockouts and overexpression line, and this work is in progress. And yeah, thank you. I would like to thank my supervisor, Dr. Hossein Borhan, and our collaborator, Dr. Kevin Rodowski, and all of my lab members and our funding agency, Agriculture Funding Consortium, and yeah, of course, Canola Week for providing us to share our work. Thank you so much.
All right, thank you for the presentation. We got one question here that's got a like up on it, so that's number one. Do you think these knockout lines may show different responses to other diseases like blackleg or verticillium, for example?
Yes, I think it should work. It should work because those genes are not like our gene as race specific. So, and this downstream defense genes, as I showed you, like we can have the broad spectrum resistance, so it should work. And yeah, we have also planned to test against them.
Thank you. We'll get one last question from the audience.
Thank you. It is really good. I have a question on the technique part.
Yes.
So the VOX, the
Yes.
overexpression, is that transient or the stable?
This one transient.
Transient.
Transient expression, yes.
Okay. So that's agro-based.
Yes.
So did you check the multiple insertion or just like the?
So multiple insertion, we have tested with the ddPCR if it's overexpressing. Oh, sorry. Yeah. So we have tested the expression, overexpression of the target gene using ddPCR.
Okay.
Yeah.
We do something very similar but different approach. So how long is the whole process from?
So it takes 4 to, like after inoculation, then 5 weeks, so like whole process it takes 6 weeks, 6 to, most 7 weeks.
Thank you.
Yeah, but this transient expression, it can be 4 to 6 weeks, can stay 4 to 6 weeks.
Thank you. Good work.
Yeah, thank you. Yeah, thank you.
Thank you so much, Medina, for the excellent and very sweet talk. Our next speaker is going to give us a break from Clubroot. Harmeet Chawla, a professor in the Department of Plant Science at the University of Manitoba, and the topic of his talk is genetic dissection of verticillium resistance in Brassica napus.
Yeah, thanks, Katie, and thanks everybody, and thanks for inviting me here. It's always lovely to come to Saskatoon and talk to the canola community here. I do realize that I stand between lunch and you guys, so I try to make it short and sweet. Okay, so I'm going to talk about our recent work on verticillium resistance in canola. So before we get into the genetics part of it, I just want to give a brief introduction or background about the genus verticillium. We have heard a lot about clubroot, but not so much about verticillium, although people are talking about it and are concerned. So verticillium genus, the last I checked, comprises of around 50 different species. A lot of them are well known. One of which comes to mind is verticillium dahliae. You might have heard about it. For Manitoba, verticillium dahliae causes verticillium wilt in potatoes, and it's a particular nasty pathogen. There's a lot of work going in that direction. Mostly verticillium dahliae is infecting Solanaceae like tomatoes, potatoes, eggplants, and etc. So other verticillium species infect species like olives and cotton. But we're going to talk a little bit about a different verticillium species here. This one is called verticillium longisporum. On the slide, you could see a microscopic picture of the spores of verticillium longisporum. And the spores are long and elongated. So probably that's where the name originated, longisporum. This pathogen infects a smaller range of hosts or a narrower range of hosts, particularly crucifers. It's pretty nasty to canola, and that's why it's getting a lot of attention nowadays. It has the potential to cause significant yield losses. Under very severe conditions, it can cause up to 80% yield losses. Here in Canada, we have heard, although not scientifically verified, yield losses of between 10 to 20%, depending on the severity of infection and other environmental factors. It was first reported in 1969 in Sweden. After that, it just took off, and now almost every European country has it. In Canada, it was discovered in 2014 in Manitoba, so it's a very good place to do verticillium research. So this 2014 discovery prompted an instant CFIA survey, and it was now, you can actually detect it pretty much everywhere. So I'm going to give you a brief reminder about the life cycle of the disease. So the disease is actually starting as microsclerotia. These are tiny spore bodies, and they are living within the roots of canola stubble, which might be left from the last season or after harvest. As soon as you plant your canola in, the canola roots will secrete some root exudates, and these root exudates will then start the germination of these microsclerotias, which would then infiltrate into the roots of the plant. Once they reach the roots, they rapidly colonize the roots, and then they want to move into the xylem tissues. So xylem is like the water transport system of the plant. And once the xylem is colonized, the fungus would live in the xylem with the plant, and it would try to clog the xylem, so it would prevent the nutrient uptake and the water movement, and it would lead into symptoms which include decaying of stem or blackening or black spots. Once that happens, the plant, of course, there will be yield losses and other visual symptoms would appear. Along with that, now the disease has fully spread out. Now, if you're a farmer, you'll come with your combine and harvest this. This will now serve as the inoculant for the next season, and hence the disease just will continue to grow. Under field conditions, there are characteristic symptoms which Canola Council has done a pretty good job at putting out on their website, how it looks like in the field. So the disease is called verticillium stripe disease, and the reason for that is it causes these long streaks or strikes along the stripe, along the length of the plant. Does not cause wilting, so that's why the name is not verticillium wilt, but verticillium stripe. It causes these characteristics gray microsclerotia, which can be used to distinguish it for other disease. One of the important points is that these symptoms only manifest very, very late in the season, so you don't see it until, let's say, right next to the harvest. So if you're scouting for this disease, then you have to be on the field right before the harvest. But it's kind of challenging. I always want to show this photo where they're comparing verticillium to blackleg. I mean, for a trained pathologist, the blackleg and verticillium might look different, but for somebody like me, for me, they look the same. So it's very, very difficult, if not impossible, to kind of distinguish these two diseases, and hence they're always confused in surveys or by people who are trying to map resistances. Not even that, under field conditions, it's also in the controlled conditions in the greenhouse that disease is not very, very simple to phenotype. Here you can see like every of those two bar plots are one independent experiment, and you have pink and blue bar plots. They represent resistant and susceptible control. The red should be lower, so the resistant should get a lower disease score, and it's the case in the first one. But if you look at the other ones, this trend kind of reverses sometimes. Like the resistant control is looking more susceptible, and the reason it happens is, so on the right side of the slides, you see the visual scoring scale, and in this scale, you could see that the visual symptoms are relying on yellowing of leaves or blackening of veins. So these symptoms usually happen in the greenhouse just from the senescing plant. So when you are actually looking at a plant in the greenhouse, you don't know whether the plant is actually just senescing and dying, or is it actually verticillium causing it. So that makes it very, very challenging to actually judge the disease correctly in the greenhouse, and that has been a recurring challenge back when I was a PhD student in Germany, and it's the same challenge we're facing in Canada now. So we kind of got our heads together to solve this problem. You know, what could we actually do to develop a better phenotyping system? And we started thinking about doing a qPCR or a PCR. So you can imagine PCR as like a photocopy machine which will actually replicate the DNA of this pathogen multiple times. So we thought, okay, we can go take a little bit of the plant sample, which will have the verticillium DNA in it. We'll push through the PCR system, and then you'll have tons of verticillium DNA. And in theory, if you detect less verticillium DNA in a plant, you can say that this plant is resistant. And if you have a high amount of verticillium DNA, you can say this plant is susceptible. So we pitched this idea to ADF, and they were kind enough to sponsor this project. I wanted to throw this slide out there. So this is the same technology they used for COVID virus detection. And if you ever took one of those tests, it costed like $300 to run one sample. And we had thousands and thousands of samples to process, although ADF gave us money, but they probably would not give us money to do that expensive test. So the project got funded a couple of years ago. In this project, we want to screen a diverse genotype panel of canola genotypes. We want to do a GWAS to identify the regions which are associated with resistance. And eventually, and hopefully, we want to develop some useful markers which the breeders can use. And of course, in this process, we have to kind of keep in mind that we want to bring the cost so more to like $1 or $2 rather than $300. So that was a significant challenge which we had in the project. So we started to set up a Brassica napus genotype panel or a diversity panel. We gave a call to the industry, to our private and public partners to contribute genotypes which they would like to see in this panel included. So we got 131 genotypes from public breeding programs, the one at U of M, and also at Sally Vale at AAFC here in Saskatoon. We included 27 lines from BNA Assist. So this is a global diversity panel, which comprises of canola accessions collected throughout the world. So we selectively only took spring types from this and avoided to have the winter types in there. And we got around 50 lines from our commercial partners, so seed producing industry here. We wanted to choose 2 locations where we can do our field trials, so in case one location fails, we don't have a total disaster in the project. We chose Portage La Prairie and Glenlee. Glenlee is the ground zero for verticillium. It's actually where verticillium was first detected, so it kind of makes sense to have that, and it's only a 15 minutes of drive from the UFM, so it's very convenient to have a location there. Portage is a bit far, but both those sites have a continuous inoculum built up, so we always have monitored these sites, and the disease pressure is quite high. So once we set up our field sites, we had a panel, we went to the field, and we planted these 200-ish genotypes. You can see a picture of my postdoc or research associate, Mohammad Yusuf, who's actually the boots on the ground and actually running the project. He seems very happy in this picture because this is at planting. So he has no clue what's coming his way right after planting. Afterwards picture, probably I'll avoid putting. So in any growing season, we go out in the field and we collect around 7,000 canola stubbles. We bring them back to the lab. We take 5 centimeters above the soil surface, and we freeze this sample as soon as we can. We freeze dry it then, and then we grind it in those containers with using metal beads. So mind you, these containers are these old film canister containers, which people, that for people my age, they would know this film which existed back in the day, and people still use it. So we go to a photo studio and source these out. Once we have crushed the samples, they are pushed through a pipeline where we have to isolate the DNA from them. We put a lysis buffer, then they are transferred into some tubes and run through the PCR machine. And finally, we have the amount of verticillium DNA which is living in this plant tissue. To give you an idea about the timeline, so we do our sample collection in August. September, we are chopping and freeze drying these samples. In October, we grind these samples. Moving on to November, we do the DNA isolation. December, we are doing the qPCR. And finally, in January, we are running the GWAS analysis. And January 31st, we report to the ADF our results. So it's kind of a very squeezed timeline where we are constantly trying to make it faster and better. Okay, so that was the timeline. Moving on to some interesting results here. This is just first year of data. Mohammad is in the lab as we speak here. He's processing the second year samples, so we'll have that result soon, and maybe next year we can come here back and present those. So here's a graph. So y-axis is the percent of Westar. So we have normalized the data on Westar. So Westar is 100%. And on the x-axis, every bar represents one genotype, which was in our panel. As you can see, Westar usually is treated as a susceptible control, but here you could see that Westar is right in the middle of our chart here. So there are lines which are better or worse than Westar. This was good for us because we have a good segregation in our diversity set. Once you do that, you have to genotype this material. So we used the 19K array. We followed a very simple, straightforward pipeline, did some filtering, plotted some PCAs, and did some GWAS, which is the next thing I'm going to talk about here. So this is the principal component analysis. For those of you who do not know how this graph works, it's just showing the relatedness or distinguishedness from one line to another. We didn't see any clear population structure, which is good for us. It's actually very good that these lines were not very related to each other. The cluster you see on the top is probably the high uracil acid lines from Rob Duncan's program, so they kind of are distinct from the other canola lines in the panel. Okay, so in any GWAS presentation, this is the picture which people are always waiting for, and maybe the rest, they really don't care about what I had to say. So here's the main result. So QTLs, we detected 3 of them above the threshold on A1, A3, and A7. My previous presenters have talked a lot about A3, so somehow this chromosome shows up for everything. So I'm going to also talk a little bit about A3 here. And we tried to focus on the region from A3. And recently there was a study from University of Alberta from Steve Strelkoff's group. And when we were looking at the significant marker on A3, we found that Steve Strelkoff was also reporting a marker for verticillium resistance very, very near to where we were. And they had a very different system, like they were scoring indoors using visual. So that gave me some confidence that we are kind of on the right track and our method is actually working and producing the results which it's supposed to produce. It's interesting that this region on A3 contains quite a lot of resistance genes for clubroot. But this resistance was not coming from Mendel. It's coming from a Swedish winter Brassica napus cultivar called Tosca. So I don't know how Tosca's background is showing up there because I have no clue about the background of most of the stuff I'm dealing with. So, but it's very interesting. And there are very obvious genes there like serine threonine protein kinase, or there are leucine-rich receptors. This region is also showing a lot of structural variation. Back when I was a student in Germany, we did a study where we saw that the leucine-rich repeat genes in these regions are duplicated or triplicated in different lines from different sources. So it's a very interesting region to look at. And kind of maybe there is a common mechanism which is occurring in those 2 diseases. And last evening over beer conversation, somebody also said that this region also has some fusarium resistance genes or susceptibility genes. So looks like it shows up for almost any disease. So it's a very interesting place to work on. Anyway, so I have 2 minutes or something, so I'd like to conclude my talk here. So qPCR method is effective in a field setting where it's difficult to judge the symptoms for the disease very clearly. People have been talking, I've heard a lot of people talking about blackleg and verticillium. So I would like to throw in a plug here saying in our assay, we just need a different primer pair. We could actually quantify blackleg pretty easily without a substantial increase in cost. So it could be a beautiful system where you could study the interaction for both the pathogens. The good news is we have good genetic resistance in our panel, so there is some light at the end of the tunnel, so maybe this resistance will finally make into the breeding lines at some point, breeding programs at some point of time. For now, we have identified 3 verticillium resistance QTL, which will be confirmed in upcoming years of studies. The QTL on A3 is close to already identified QTLs for verticillium and co-localizes with the hotspot for clubroot resistance, so something to think about. 2 more years of field testing data coming. 1 year is we are doing it right now as we speak, so hopefully we'll have some very, very interesting results for this year, and we'll continue doing that at least for 1 more year. With that, I'd like to thank a bunch of people who actually do the work on the ground, and I get to stand here and present it. So those are the real heroes of the project. And of course, none of this work will be possible without the support of the agriculture development funds from the government of Saskatchewan. The project is also supported by Sask Oilseeds, WGRF, Manitoba Canola Growers, and it's supported through the Sustainable Canadian Agriculture Partnership. My email is out there. I'll be here till the end of the day. If you do want to chat something about verticillium, more than happy to chat. Thank you very much.
Thank you, Harmeet, for the presentation. One question here. You made note of multiple clubroot resistance found on AO3 in that area. Is it possible that these are the same, but just annotated separately by different groups?
There has been, like, so the paper I was talking about where I was involved and we were looking closely at this Tosca region, that, like, there are some, the Tosca resistance is very distinct to Mendel resistance, although they're very close by. So there is incidences where there are similar genes and annotated twice, but there is also incidences that there are two different genes actually. So Mendel is around 14, I want to say 20-ish MB, 20, 25 MB on A3. Tosca is around 10, 15. So there's like a 10 MB gap in between them.
Thank you.
Thank you, Harmeet. Excellent presentation. Is there any chance to include Topaz R11 line, because that is our new WCRRC verticillium check?
If you can provide the seeds, we can include it. I think in the first year of data, we did had a bunch of Topaz, but I don't know if it was the exact line you're talking about. But if you can provide us the seed, we'll include it. Yeah.
Okay, well, thank you.
Yeah. And also for other industry members, if somebody wants to provide some more lines, we still have some capacity. So please see me if that's something of interest.
Thank you very much, sir.
Yeah, thanks, Kathy.
Thank you very much to Harmeet and all of you for the excellent questions. This session was sponsored by Genome Prairie. And we have a few minutes to listen. Where is Mike? Oh, there he is. to listen to Mike say a few words, and thank you very much for the sponsorship. After that, there's lunch, and thank you very much, everybody.
Man, to be the one really standing in front of lunch. I'll keep it short. Thank you, Kathy. I am Mike Say. I'm the CEO of Genome Prairie. Many of you, I think, would know of us. Maybe some of you don't. We're very pleased to be here. Just very quickly, Genome Prairie is part of the National Genomic Enterprise. We've been at it for 25 years. Genome Prairie covers Manitoba and Saskatchewan. We're one of 6 regional centers across the country, and it's been structured that way and worked pretty well in a competitive fashion in that we can represent and advance the regional priorities at hand in all things genomics. Canola is probably the best example of a national interest piece of our economy that is fairly regionally specific. Certainly does serve the national interest, given canola is more than 15% of the total agri-food exports across the country, and that's over $100 billion now. So I will say on the national interest piece, our sponsorship of this event probably demonstrates that pretty well, in that when Genome Prairie was approached to act as a sponsor, and Wilf approached me, and as we all know, no one says no to Wilf when you get approached. I did reach out to our colleagues across the country, so this isn't just a sponsorship of Genome Prairie, and we quickly received the support of Genome BC, Genome Alberta, Ontario Genomics, and Genome Canada. The opportunity still exists with Genome Quebec and Atlantic Canada to grow our canola presence, but I'd certainly like to thank and recognize the other regional centers and Genome Canada for supporting in that sponsorship. And in my mind, that's just a demonstration that even though canola is, yes, mostly regionally grown on the prairies, it is certainly recognized as something that is in the national interest to support. I just want to take a quick moment too to share with you a new program that we launched this year, and it's called GenTrack. We love our acronyms. GenTrack is the Genomics Translation and Commercialization Initiative. Many of those that would be familiar with our major projects that we've funded over 25 years, they're typically larger scale, multi-year, and they are vetted and approved at the national level by Genome Canada. GenTrack is a little bit different in that we've evolved, in that the focus is on commercialization. And while we still will be looking for academic research leads on these projects, there is a requirement that there's a commercial receptor at the other end of these, and that is new for us. The other thing that is new is that the decision making on approved funding for projects was typically conducted by Genome Canada. We've now localized that, so funding decisions will be made at our level, at the regional level. So if you're interested in those programs, I'd invite you to have a look at our website. Lester Young is our Chief Science Officer, and Rebecca Carroll is our Manager of Strategic Innovation. So we're excited about that. This is a 4-year program that'll be funded annually, and cost-shared program. We had good uptake on the first program. We'll be making funding decisions actually at the end of this month, and we'll be putting out a new call again early in the new year. So thank you again for the opportunity to participate. We thank again our sponsoring partners in Alberta, BC, Ontario, and at the Genome Canada level. And thank you to all of you, because canola doesn't maintain and improve without the efforts of everybody in this room. So thanks again, and now enjoy lunch.